Hi there, and welcome to tonight's webinar, Casting a Wider Net for DNA. This is a follow-up to the presentation I did, I think a year ago, a little bit more than a year ago. So check that out on part one on Friends of the Carp Hills website. You should be able to find a link. So this presentation tonight is about environmental DNA and what we did for collecting environmental DNA in the Carp Barrens. So the order for the presentation is really, I'm going to talk a little bit about the motivation, why I decided to embark on this journey, a little quick biology 101, talk about some of the results from this year, and then talk a little bit about what's next. But first off, I really want to thank the donors who, donated to this project, and not only who donated, monetarily, but also who just encouraged me, sent me emails, positive feedback on the work we had done previously. So it means a lot, keeps us going. The bottom right inset picture is just, I'm holding a completed kit, and we'll talk a little bit about what the kit is and how the process is done. So for me, the motivating scientific question around doing environmental DNA for the Carp Barrens was, is it possible to catalog a pond's ecosystem using molecular biology? So, just will we learn things we didn't know already? Things that we have a lot of data on iNaturalist, for example. And also, is there value in knowing these things? So yeah, let's just take a closer look together. So just to situate these are the ponds in the Carp Barrens. If you're not from the Ottawa area, just really quickly, this is between the nation's capital, Ottawa, and Carp, a small town, and a very nice natural area that's situated between the two of them. So no urban development immediately within the Barrens, but on both sides. So we have 4 locations for the ponds in the Carp Barrens, colloquially known as Peggy's Cove. And then we have the Fen Pond and the Rockwall Pond. The difference between Peggy's Cove 1 and 2 is one of them has a greater water flow to kind of a connecting pond between the two of them. So that was the sampling that was done in 2021. I'll be talking a little bit about the results from that pond in a little bit. I also did some sampling in August of 2022 at the Crazy Horse Beaver Pond. This is a trail that is a few miles away from the Carp Barrens. And those are just some pictures I took on that day at the pond that I did sampling of. And the check marks are actually the species that were identified by environmental DNA. The gray heron, I believe it is, or it was not picked up. I may be wrong on that. Maybe a blue heron, not sure. Okay, so just a little bit of background. I'm not a biologist, as you could tell. I couldn't identify the heron. But anyway, the stuff that I do know is all centered around environmental DNA. So DNA is found in all living or once living things. And environmental DNA are really the small piece of the DNA that we find in nature. So, as humans, we shed 100 million skin cells a day, and each one of those small fragments will have some DNA within it. When animals or amphibians or fish or other things, worms at the bottom of the pond, when they interact with the water system, they all shed some DNA. So DNA, it's kind of famously known for the double helix. There's about 2 billion of these letters, A, C, T, and G, in the human genome. When we're talking about eDNA, we're looking for small fragments of small sections within specific genes. So we're targeting maybe 300 letters out of billions of letters. And we're doing this using special chemicals, chemical means to do this. But main thing to take away is eDNA are small fragments found in nature, and those fragments are going to allow us to identify specific creatures. So the entire eDNA process really is 8 steps, and we only actually really do number 1 and number 8. So, we go out and we collect water from the ponds through a special filtered system. Funny enough, the picture shows somebody without gloves. We would always be using gloves. That's not any of our researchers doing that. And then what we'll do is we'll extract DNA from that water body, and then we'll amplify those specific regions that we're targeting for the species that we're looking for. And there's quite a bit of complex computational work, a whole new field of bioinformatics. And then that data from the lab, in this case, it was Jonah Ventures, they send that information back to us with a lot of identification information in it. But then we, as I'll say, curators or amateur biologists, need to try and figure out, do those identities make sense for our area? And I'll cover that off in quite a little bit more detail. So our collection methods, as mentioned before, really simple. You can see Mary Gwen, a biologist there. She's just sampling water from one of the ponds. You can see that's quite, it's like a tea-colored pond, but that's the way the Barrens ponds look. And we squeeze the water through this filter until it just drips out at about 1 drip every 2 seconds. So the filter gets quite clogged. You can almost see the brown color there of the filter as we're pushing it through. And then we take the filter off carefully and we inject some preservative through it, cap it, seal it, and FedEx it to the lab. So it's fresh and ready for sequencing. So a little bit more about the how, those pieces between 1 and 8. So we're going to amplify these small regions. We colloquially call them barcodes. And these are the sequences that we're going to sequence that DNA, the ones that we amplified, and then we're going to identify that DNA using databases. So amplify, so just take that amount of DNA. Every cell has about 1/trillionth of a gram. We're going to amplify it up to a millionth of a gram. And then we're going to send that to a lab. They're going to sequence it. We're going to sequence this back, and then somebody's going to identify those sequences against species. And I'll talk a little bit more about that as well. So you can think about the barcodes, very analogous to what you'd see at the supermarket. So we got our Quaker Oats, we scan it. It's got this UPC code. Well, when we get a DNA sequence back, you'll get a number of letters, as I said, under 300. And those 300 letters are in a specific gene region that we target for plants, for example. And they will resolve in the database to a species, hopefully a species, but not always. In this case, that combination of letters actually does resolve to that variegated euonymus fortunei. Now, sometimes, and you'll see it in the results, we're looking for X and we find Y. So, we're looking for fish and we find beavers. Now, we do count these off-target matches if they fall into our criteria of greater than 97% DNA match. So the important thing is then when we get the results back from the lab, the level of matchingness, I'm going to use that word, varies. So in some cases, we have 100% match with a reference sequence in the database. And then we're, very confident that that species was found there. Or a species that's very close to it. And then we say, okay, we're going to count that as a species within our pond. But you will see many off-target matches in our DNA. And I can get into the technical reasons why that is. You can send me an email at dna@carphills.com and I'll tell you more about those reasons, or if you have any other questions. So one thing we want to do is that we want to identify organisms at the right level of classification. So I have some handouts and I can again send them to you if you're not in the live audience tonight, if you're just listening to this. But there's a sophisticated bioinformatics to classify these at the right level. So typically, in my dream when I started doing this work was, oh, if I'm just going to take a little bit of water and I'm going to push it through the syringe, I'm going to send it off to the lab. The lab is going to come back and say, oh, you have these 300 species. So that was my desire. But the reality is that the databases are maybe incomplete or they may be data missing. So we only get the resolution at the genus or family level. And that's not nothing, but it's not, you know, the very, very specific species level identification. Okay, so what did we do and when did we do it? So we did collect 1,457 unique DNA sequences over the last 2 years. It's 3 years, I should say. It started in 2011. It's 2021. And most of this data was previously presented, so in January 2022. But these other ones I've never presented before, so those are the ones I'm going to focus on tonight. And I'm also going to focus on some comparisons where we had comparative data, so fish and macroinvertebrates between a couple locations, and also year-over-year data for a couple locations as well. So, you know, was there a big change in the composition for Peggy's Cove 1 between 2021 and 2022? Well, we can find out. Okay, so let's start looking at some of the data here. So, and there's a lot more data on different genus and species, but I just picked a few representative ones just to illustrate environmental DNA here. So at the species level, we found American bullfrogs in Peggy's Cove 2. And at the genus level, this might be a leopard frog or green frog. And again, I'm not a biologist, apologies. But we don't really know at this level of species whether, you know, these were actually found. We just have we know for certain, though, this genus was found. And this is the relative DNA abundance, the number of amplicons. So, you know, 40 times as many frogs were found, but we couldn't resolve those to be, American bullfrogs. So these are the two sequences that came up. And this one is 100% match for an American bullfrog. And this one is a 98% match for multiple species of lithobates. So we don't actually say it's these species. Instead, we say there's lithobates in that pond, and quite a lot. We did do some tests for aquatic plants, and we did find some aquatic plants, but I'm just actually going to focus on some other kind of off-target plants that showed up here. So at the family level, we found heathers at the rock wall and Peggy's Cove. And a good example heather here is the leatherleaf. Janet Mason donated the picture, and I'm not going to try and pronounce the Latin name, but you know, we'll pick them up because leaves may have fallen into the water. One of the things I didn't mention is that environmental DNA can last quite a while in water. So people will ask, okay, you know, how long does the DNA last in the water? Can last anywhere from, you know, 7 to 10 days to 2 weeks, depending on conditions. So sun degrades DNA, temperature degrades DNA, but you can definitely pick up things that have been in the pond or interacted with the pond or died in the pond for a significant period of time, not just like an hour before we show up. So aquatic plants, again, at the genus level, we found some mosses and some pondweed. So again, a nice example here from Carp Barrens. But in the Vicinium family, which is, the heath family, we don't know whether it's blueberries or some other berries. So, these are kind of examples of potential plants that could have been in those ponds. So at the species level, again, we're going down that pyramid to things that are, definitely there, that have sequences within the database. Common water shield was just massive within most locations. And then these two other species of algae were, 100% matches. Bacteria was kind of really interesting to me because, bacteria are the decomposers of the ponds, right? You have, if I go back here to algae or aquatic plants, they're taking CO2 in and they're producing oxygen and nutrients. Well, bacteria, for the most part, are breaking down the dead things and producing CO2. So that's kind of a fascinating interplay between these species. The picture on the right is a very long bacteria name. And when I originally saw it, I was reading about some of these bacteria, and it's one of those bacteria that doesn't react well to antibiotics, so antibiotic resistant bacteria. But it's very common in soil. And one of those reasons you wouldn't want to drink the water. Not only that, there's lots of Enterobacter bacteria. So that's like E. coli in that order, and Salmonella in all the ponds. So you don't want to do that. One of the interesting observations, at least for me, was that there were some methane eaters in the pond. So some different families of bacteria that eat methane, and in the Carp Hills, you can sometimes smell the methane from those ponds. So if those families weren't there, we'd probably smell those ponds quite a bit more. Okay, so a little bit of comparative data, and this is really actually where I just wanted to point out as kind of a new environmental DNA biologist in this field. It's kind of new in general. I think my sampling techniques failed me. So ponds, as we all know, are fairly still bodies of water, right? Almost nothing, no water movement in a pond. But when I was doing my sampling, I would only sample in like one location in a pond. But so we don't see a lot of, green here because I was only in one spot in the pond. So this coming summer, when I'm going to be sampling, I will traverse the pond and take lots of water samples, combine them together, and then push them through my syringe to extract the DNA. I did do some sampling over 2 years in a stream, a nearby stream, not part of the Carp Barrens or the Crazy Horse, and I had 6 species of fish, and all 6 species showed up in the second year. So I'm confident that if I improve my sampling techniques, I'll see more green here in future years. Okay, so again, different locations over 2 years. Probably more a example of me not sampling in enough spots, but picked up some rotifers here. These guys are the garbage collectors of the pond. So they're decomposers like the bacteria, just a lot larger than the bacteria. Sorry, I just noticed there's some mayflies, right? Those are the things that indicate good pond health, one of the indicators. So some fish, comparing 2 locations. So I said I'd compare Peggy's Cove and Crazy Horse. Well, there was 3 fish species that were common across both, but some notable off targets, right? So I picked up that wood duck that I'd shown in my first picture, picked up American beaver. Would like to pick them up in the beaver pond, but that didn't happen. Painted turtle, picked them up in the beaver pond, which was great, and eastern newt. And I will remark that other than the wood duck and the painted turtle, I didn't see any of these other species that I picked up, especially the newt. So that's really, a nice example of where environmental DNA allows us to find things that we aren't seeing. Peggy's Cove and Crazy Horse, again, not a lot of commonality. Macroinvertebrates, because a lot of them have hard outer shells, don't shed DNA, not like, slimy fish. When a fish is going through the water column, well, first of all, it's moving a lot, and it's also, shedding a lot of DNA as it moves. So these macroinvertebrates, not the same, and except for the species of algae was picked up in the macroinvertebrate test. Again, like I said, an off-target hit. I had funded some special tests for reptiles and mollusks, which were a bust, sadly. Those specific tests. I did pick up the painted turtle, as I mentioned before, and I picked up pond snails, which was awesome. But I picked them up not on the reptiles test. I picked them up on off-target tests. So now mollusks themselves, specifically, like we did not want to pick up, or didn't expect to pick up zebra mussels in a pond, and we're happy not to have invasive mollusks in the pond. So, not necessarily bad that the mollusk tests were a bust, but the other one was disappointing. We didn't pick up any Blanding's turtles, for example. One of the interesting things is that you get a lot of data back. I think on my first slide, I said we, get almost 1,500 DNA samples, and it's really hard to process all that data. So I was interested in the pond system interaction. So how do all these organisms interact? And I found ChatGPT to be an excellent research assistant. So I would send it in lists of species, and and ask for the relationship in the food web, and it would tell me about them. So the saying there, big fleas have little fleas upon their backs to bite them, and little fleas have lesser fleas, and so ad infinitum is a kind of a biologist saying that, there's always somebody in the food web higher, generally speaking. So this is just a single pond view, system view of the primary producers, like I talked about before, the algae and the things that, have chlorophyll in them. They're taking that sunshine, they're taking those organic components from the decomposers, and they become those primary producers. So they're taking in CO2 and organic compounds, outputting essentially sugars, other organic compounds, and oxygen. So thanks ChatGPT for characterizing these species for me. And then, at the bottom of the food chain, if you will, the decomposers are breaking down those dead items and, turning them back into more organic compounds and emitting CO2. So as I said, ChatGPT was a great research assistant. In the previous year, when I was presenting this data, I didn't have ChatGPT, and I really could not do the family and genus level research, nor could I do the food web analysis. So, and it did help me too with some basic quality assurance. So, sometimes you'll have a species that'll come back as a match, but when you are doing a validation, you realize it's a 97% match, but it's a saltwater species or a tropical species. That does happen. So you can't just blindly take the DNA data and just say these are all the things that are there. That's just a little bit, it would be a little bit naive. So this is just a sample interaction with ChatGPT, and it does, you can see I've provided you a list, and you've provided me analysis like this. Oh yes, I remember. So remembers your previous conversations and its language models, and you can ask it to produce tables and provide the URLs, for example, of where's the data, the source of the data that it's giving you this answer for, so you can go and check it out as well. So just a couple general findings here. I guess no surprise really. eDNA seems to have better species level accuracy for larger organisms. I think that's natural, right? Because biologists can go pull up a hair, this little beaver here, and they can sequence that hair. Whereas for bacteria and really, really small organisms, it's very hard to isolate them and then sequence the DNA. And there's maybe not as much interest in general in those smaller species. I maybe didn't mention it, but that we did in 2021 was in October, whereas the sampling from last summer was in August. So really dramatic difference in the abundance of DNA across those two time frames. Interestingly, the Ottawa Riverkeeper shared some of their data, and I guess not surprisingly, the ponds and the rivers are quite different in terms of their species, the ones we found. Now, it's not a like for like comparison because they didn't necessarily run all the tests that we did. I don't think they did bacteria or macroinvertebrates, but for the tests they did do, there was only 5 out of 60 species that were common. One thing that's interesting, and I'll talk a little bit more about this at the very end, about challenges around doing environmental DNA for complete pond inventory, is there are a lot of sequences that are actually completely unknown. So 254 sequences are not yet known. So over the next few years, we'll just continue to take those sequences to back to the databases, reference databases, and ask, do you have an answer for me now? As the research suggests, eDNA picks up species that aren't easily observed. So, bacteria, for example, and aquatic insects, and so on. So yeah, that's the recap of all of the information. There is a lot more species data. I'm happy to share that with anyone who wants it and other questions you might have. So you can just send me an email at dna@carphills.com. So to recap the motivating scientific questions. So can we determine the entire pond ecosystem through molecular methods alone? So environmental DNA specifically, metabarcoding. So no, I don't think so, not at this time. So there's some gaps. So first of all, even just within our ponds, we had 17% unknowns. And the other one that is a pretty significant gap is quantification. So eDNA is never going to replace, field biologists and people observing, the real live animals. So we can't do quantification right now. So, we didn't go fishing for DNA, but there are holes in the net, clearly. Things are not being captured, and sometimes we're capturing things that we don't expect to capture. I talked about those off-target species. So really, I guess the question that remains, is should we stop fishing? I thought a little bit about that, and I'll cover that on maybe the last slide. But before I go there, I'll just talk about what am I going to do next? So I'm just going to continue doing the sampling. I will continue my molecular biology studies. I'm going to sample the ponds in 2023 every so many feet, and then I'll save those samples until I have sufficient funds to sequence them all. Drone sampling is something of real interest to me, but that's a whole other level of expense. But I think it is probably the way a lot of people will go for, say, very commercial uses of this technology. So just to kind of return onto this question, is eDNA worth continuing? Yeah, I think it does serve possibly a very important purpose. The world is very data oriented today, and you're basically creating a body of data, body of knowledge about the carp barrens and the Crazy Horse Trail conservation area. So, it's hard to prove there's change if you don't have, a data set that, pre-change, right? It gives a lot of insights into unseen and difficult to observe species. So over time, I think we're going to see better, producer, decomposer, consumer ratios and DNA-based indicators of health, slowly getting there. And if the water samples are stored properly too, they could be re-examined for many years. So as the technology improves and the primer sets of things that isolate specific DNA, as that improves, they can be re-examined for many years. One of the other important things too is the ability to contribute to core science research, right? So I think that's an important part of doing environmental DNA. We could be discovering new species and contributing to the, knowledge, the science, the knowledge of science. So I already mentioned the last 2 points. Something changes in a pond, we'll have a baseline, and then we have an evidence base for conservation. Tangentially, there are some global trends that there's a lot of investment and interest in eDNA and nature-based accounting. So, I've heard people talk about gross biodiversity indicators instead of GDP or gross domestic income, talking about gross domestic biodiversity. So there's really some interesting things happening there. So yeah, I think it's a great area. I think it's important, and I will continue doing it, and I hope that I will continue to do it with the support of the Friends of the Carp Hills. And that's about all I have for tonight. Any questions?